So, hello everyone. We are here once again in the what we are reading section of the South American Mycorrhizal Research Network. This time in June 2021, and we are delighted to have Mathu Malar. She is a postdoc in the University of Ottawa, and recently. Um, she published this article in Current Biology entitled The Genome of Geosiphon pyriformis reveals ancestral traits linked to the emergence of the arbuscular mycorrhizal symbiosis. It's an extremely interesting article. Madhu, tell us in the first place how this article came about, in which context, in which project or collaboration.
Thank you, Sisa, for having me in the meeting. And so this project, it is all about the Geosyphon pyriformis genome sequencing, whereas the Geosyphon is one of the endosymbiont with the cyanobacteria called Nostoc, and it is thought to be originally, it is thought to be lost. But our collaborators, Manuela and the Claudia Kruger from Germany, managed to rediscover it and contacted my supervisor whether he would be interested in sequencing the genome on that. And this genome data was like available even before I joined this lab as a postdoc. And the producing the genome assembly was extremely difficult because this geosiphon lives with the comes with all other soil contaminants and all other fungus and everything. So I used a metagenomic based approaches to use to get the clear assembled genome. And once I got the decent genome assembly, and each individual collaborators have joined to provide their expertise on the phylogenomics and host symbiosis related association studies.
Perfect. Tell me exactly, you mentioned that you did some metagenomic analysis, um, and I understand that you originally are like a bioinformatician. But this paper goes deep, very deep into fungal biology and fungal evolution. How has been this transition for you from a career perspective?
I do come from a strong computational and the bioinformatics background. While I was in my PhD, I got fascinated about the fungi because of their complex life cycle and the mating system. And most of my knowledge was centered towards the genomics as opposed to be the biology. So I contacted Dr. Kurati that whether I can join his lab as a postdoc, and he agreed. Then as soon as I arrived in the lab, he was kind enough to share some books on the fungal biology, and he explained the complexity of the AMF multinucleate system. That built an excitement around the Geosyphon project, and I am just happy to be involved in this amazing research.
And why is Geosyphon so interesting? Why is so special? And why maybe its distribution is so restricted?
Geosyphon is the only fungus which forms endosymbiosis with the cyanobacteria called Nostoc. And this forms a complex bladder like structure, and it contains the Nostoc cells inside. So far, it's been only found in the Germany. It is possible that it might be found somewhere else. And also, like I would say, like people have not searched hard enough to look for it. Also, culturing it in the lab is like very difficult because it requires maintenance of the constant humidity. This is the one of the reason it might have got lost.
Okay, and tell us, in general, in the article you mentioned that you found like more than 24,000 genes in the genome of this fungus. Tell us what else is interesting in the genome. of this fungus?
The number of repeats and the genes are similar to that of the other AMF. The same set of genes are present like other AMF. Similarly, it can do the same function like other other arbuscular mycorrhizal fungus. So this is like a good indication that Geosyphon might rarely undergo the formation of the mycorrhizae.
And what about the like phylogenomic analysis you did? Uh,
So,
in,
the phylogenomic analysis have been performed by my collaborators, Yan Van from University of Toronto and Jason Stadich from the UC Riverside. Originally, people thought that Geosyphon is an ancestral lineage of the AMF, but it is really found to be the one of the early diverging AMF clade, such as Paraglomus and the Ambispora.
I see. And also in the article you mentioned that you compared this genome with other five genomes from glomeromycotina and four genomes from mucoromycota. Um, what were like the main conclusions of doing this comparison? Um, can you tell us about this?
Yeah. Before our study, it was shown that AMF have lost like genes which are responsible for the production of sugar and the fatty acid synthesis. And the genome of the geosiphon also showed that those genes are also not found in the in this early diverging AMF. Altogether, like this, all my results suggest that these genes are like lost even before the emergence of the glomeromycotina clade.
And which are the consequences of these losses?
The consequences are, these genes are like lost even before the emergence of the glomeromycotina.
I
The
see.
obligate biotrophy process has been like there even before the, the formation of this clay, glomeromycotina clay.
Um, you also did some analysis involving a very common AMF species like Rhizophagus irregularis. Um, which were the main conclusions when comparing with Rhizophagus?
So the comparison with the Rhizophagus irregularis have confirmed all like what all others have found before. And I also like found this genome is like homokaryon, and it has lost all the genes necessary to produce the sugar and the lipid.
I see. And from a point of view, one of the most interesting things on this paper was all the things related to the Nostoc symbiosis. Um, you say that you produce evidence of horizontal gene transfer between Geosiphon and Nostoc. Can you elaborate on this and how this horizontal gene transfer works?
Yeah, we like there was a horizontal gene transfer of happening between the Nostoc and other bacteria also. Like that, we have identified 18 genes having the gene horizontal gene transfer, which is likely to be derived from the bacteria, including from the Nostoc. And Nostoc and the other endosymbiont bacteria coexist in the bladder. During the coexistence, there is a chance of the genetic exchange between the partners. This highlights the importance of the horizontal gene transfer for the fungal evolution.
Perfect. Well, I think that we can start to wrapping out. Something else that you want to say, a final word, like a final conclusion about this article, or maybe invitation for other mycorrhizal researchers?
Yeah, thank you very much for having me in this interview. So the final take-home message for this project and my research, I would say like this genome information is very helpful for the researchers, those who are working on the AMF. And it is helpful to understand the symbiotic relationship, and it paves the road to study this fungus using the tools of molecular biology. Yeah.
Perfect. Thank you very much, Matthew, for this wonderful interview. And I hope to see you next for the next interview of the South American Mycorrhizal Research Network.
Yeah, thank you very much, Cesar, for having me.
Thanks. Bye.